Thanks for joining us for another episode of Biotech and Life Sciences CEO Stories. Our guests today are Doctors Jonathan Arambula and Krystle Karoscik, co-founders of InnovoTex. InnovoTex is an oncology therapeutics company developing a platform of next-gen small molecules capable of circumventing drug resistance, often seen in standard of care treatments for advanced solid tumors. The InnovoTex technology is licensed from UT Austin and MD Anderson Cancer Center. The innovation is a next-gen antibody-drug conjugate-like platform that uses a clinically validated tumor-localizing macrocycle to create TDCs. These novel TDCs deliver anti-cancer therapies or payloads directly into the tumor. Other co-inventors of the technology, Doctors Jonathan Sessler and Zahid Siddiq, continue to be integral collaborators and lead the scientific advisory board. Dr. Arambula has held the position of CEO and been a member of the board of directors since May of 22. Previously, he served as vice president of research, then CEO at Oncotex from November of 19 to September 2021. Dr. Arambula is a world-leading scientist that has over 15 years' experience in drug discovery and development of cancer therapeutics. He's also a co-inventor of Oxytex and the TexCore technology that is now being developed by InnovoTex. Dr. Karoscik has held the position of COO since May of 2022. Previously, she served as Director of Development Operations at Oncotex, Director of Portfolio Management and Operations, Clinical Trial Operations Lead and Strategist for clinical research organizations, and fractional executive positions for early-stage therapeutic development companies. She holds dual master's degrees in neuroscience and bioethics and a doctorate in entrepreneurship. She's the recipient of numerous Innovation Excellence Awards for the development of therapeutic infrastructure programs within the university ecosystem. So, InnovoTex is developing texaferin drug conjugates, or TDCs, to overcome standard of care resistance. What's texaferin, and what are TDCs?
Yeah, no, absolutely. Yeah, no, absolutely, Tim. Yeah, thanks for having us, by the way.
Sure.
So texaferins are a macrocycle. Okay, so macrocycles, the most common one that we're likely aware of is heme in our body, which gives the red blood cells, well, the red color. So texaferins are essentially an expanded heme molecule and sort of in the shape of a donut. And in the middle, you can actually incorporate large metals. And so with texaferins, what you do is you can have a large metal in the middle, and it can render it MRI active. So you can actually monitor the tumor localization of these unique expanded macrocycles being delivered directly to the tumor. So in previous studies, texaferins were illustrated in patients to localize to metastatic lung cancer that metastasized to the brain. They were able to see it at a ratio of 70-1 relative to normal or non-tumor tissue. So here at InnovoTex, we're utilizing this macrocycle as a delivery vector for various anti-cancer payloads. The main reason is that it's a clinically validated tumor localizing agent. So just to give a little backstory of what texaferins are and what they've been through phase 3 clinical trials, and they've been established or well validated in over 1,000 patients to have that very efficient tumor localization property. So what happened is that now InnovoTex has had the opportunity to license the entire pre-existing IP portfolio for not just texaferins, but newer texaferin drug conjugates, or TDCs. And that's what we're working on right now, is developing our lead clinical candidate, Oxalotex, for the treatment of solid tumors.
Okay. So what's the text core platform? What can just trying to get the
Absolutely.
definitions out of the way.
Yeah, no, absolutely. So TextCore is our discovery platform. It's basically our discovery platform that allows us to incorporate that tumor localizing vector TD or Texaprin into our TDC development. Our first lead clinical candidate is known as oxalotex. And so that is in preclinical development. So on the verge of embarking on the IND-enabling studies, and we're using that as our proof of concept for the platform, for the discovery platform.
Jonathan, I noticed in my work preparing,
Mhm.
you had earlier worked on this technology platform at in another way, in another venue.
Yeah, at a different company. Yeah.
Yeah. Can you kind of outline a few of the challenges that you experienced in terms of development and commercialization, that sort of thing?
Yeah, so originally this platform was licensed in 2019 to a large Australian biotech conglomerate. Of course, this is pre-COVID. And I actually joined the team as initially as VP of Research, and they spent about $6.5 million in development to move it closer to the clinic. Of course, this is pre-COVID, and because of the COVID lockdowns, especially the ones that occurred in Australia, the company essentially ceased operations. And so the technology was given back to UT, but this provided us with a key opportunity. One, we worked very closely or diligently with UT inventors and some prior investors in that company to be able to form that new corporate entity InnovoTex to continue the development of this technology.
So where are you now in the development process?
We're on the verge of finishing up our, I guess you would call it the pre-IND enabling studies. And
Mhm.
then once we're done with that, we're moving into the IND enabling program.
Okay. And what would be the proposed target indications for oxalotex?
Oh, no, absolutely. So oxalotex is being developed for solid tumors that suffer from platinum drug resistance. But what we have found is that it's a very unique and specific biomarker profile. So it needs to have a wild type P53, but it has to be these tumors that we're efficacious or we're proposing to treat need to have platinum drug resistance. So here's the issue that we're dealing with. So the clinical utility of platinum uh, is limited by what we call either intrinsic or acquired resistance. Okay. So they could be de novo refractory to the platinum treatment, or they can build up acquired resistance. And so this impacts that patient population. This is exemplified by a 5-year survival rate of just roughly around 27%.
This is
So
in ovarian cancer specifically, or
no, this
across
actually
all?
occurs across a variety of different solid
Right.
tumor types.
Okay.
This has been observed in a variety of different solid tumor types, but we're using ovarian cancer as our proof of concept, mainly because it is an orphan disease. And we have a significant amount of data illustrating the high utility of oxalotex for ovarian cancer, or at least those ovarian cancer types that are wild type P53, platinum refractory, or resistant.
And would this wild type P53 issue exist across multiple solid tumors, or right?
Oh, absolutely. Yeah. So when you look at the data across a variety of solid tumors, ovarian cancer has a substantial about, I would say around 40%, 35, 40% are wild type P53 platinum refractory or resistant. There are other larger markets that have a much more substantial number of platinum resistance as well.
Mhm.
or wild type
Do you have
P53
a feel
platinum.
for, do you have a feel for how large the market would be just in that ovarian cancer subset?
Yeah, so it impacts around 300,000 women on an annual basis globally. And so if you take that 300,000, it would likely impact anywhere between 100 to about 130,000 women.
Wow. Okay. And that would be, okay, good. So tell us more about the platinum resistance. Why is that important?
No, absolutely. So traditionally, platinums such as cisplatin and carboplatin, they are designed in a way to damage and target the cancer DNA. Okay, so essentially what happens is that once they damage the cancer DNA, it goes through cell cycle arrest, it tries to repair itself, and if it doesn't, it'll eventually activate P53 and undergo apoptosis. Okay, or cancer cell death. That's their traditional path of going from DNA damage to cancer cell death. So what we have found, or actually it wasn't us, what was discovered in 2017 by the Stephen Lippard group up at MIT is that not all platinums are created equal. So if you design a platinum in a way that is a little bit greasier, a little bit lipophilic, it actually does not necessarily induce a DNA damage response, but rather it induces a ribosome biogenesis stress response. So it targets a different portion, or what they were proposing was it targets a different part of the cell. And by doing so, we were finally able to explain the cross-resistance profile differences between cisplatin and something that's a little bit greasier, such as the platinums that are used for GI cancers, such as oxaliplatin. So what we decided to do in our design methodology was to incorporate a different type of platinum into our TDC to be able to target the ribosome to be able to circumvent this P53 issue.
Okay. And so, of course, ADCs, antibody drug conjugates are incredibly popular now in the industry.
Absolutely.
Everyone's
Yeah.
going after that particular thing. Why do you pursue, in light of that, TDCs, and how do they differ from ADCs?
Right. So this kind of stemmed back to when I started as a postdoc at University of Texas at Austin. I was an American Cancer Society postdoctoral fellow back then. And when I started in the lab, we had this crazy idea to where we could utilize texiferin, which was invented in that laboratory as well,
Okay.
in which we could utilize texiferin as that drug delivery vector to be able to deliver something directly to the solid tumor. The innovation that came from the Zahid Siddiq lab at MD Anderson Cancer Center was what type of payload to incorporate. And so we were able to collaborate together to create this novel innovation that InnovoTex now has the license for. And so by targeting the ribosome, it allows us to circumvent all resistance factors that are typically associated to that wild type P53 platinum resistance. We see full activation of that pathway and complete circumvention of all resistance factors associated to traditional DNA damaging agents, which are typically used for ovarian cancer. In fact, this agent was so potent, it was 25-fold, it had 25-fold greater potency than the original macrocycle by itself.
Wow. Okay.
Mhm.
Um, So you've shown kind of an increased therapeutic benefit and improved safety levels versus oxaliplatin. Why is why is it better?
The short answer is safety, really, and what's actually used in the clinic. So remember, oxaliplatin is a tumor localizing agent. It's also a prodrug. Okay, so
Tell us
so
what is that? What's a prodrug?
A prodrug is something that delivers an anti-cancer agent that is in a different form from its active form. So the texaferin is actually delivering an anti-cancer payload, but it is not the actual payload that will actually kill the cancer. It goes through an oxidative reduction to generate the active form in the cancer. So we get two different benefits right here. So oxalotex is a tumor localizing agent in a prodrug form. So we get the localization benefits from the texaferin core or from that macrocycle.
Okay.
Then we're also delivering a prodrug version of the active form of the payload. So once it delivers, so it's essentially inert or less, it illustrates fewer adverse events while it's being delivered. So once it gets to the cancer, it's able to release the payload in its active form, and that's when you start to see, well, essentially cancer cell death.
Okay.
So we illustrated this just in a simple study a couple of years ago. It was a side-by-side safety comparison between oxalotex and the FDA-approved platinum known as oxaliplatin. We did a safety study in mice, and because oxalotex is in a prodrug form, we're able to dose a significantly higher amount. There's a much greater tolerability of oxalotex. And so when we dosed at a much higher amount in PDX efficacy models, we actually were able to see a substantial amount of efficacy against the solid tumor in a model that was essentially completely non-responsive to oxaliplatin when we dosed it at its MTD. So what we wanted to do was see if we could increase the dose of oxaliplatin to see if we could see any sort of glimpse of efficacy. So once we increased the dose to the standard dose of oxalotex, unfortunately, about 40% of the mice died from oxaliplatin because of those adverse effects. You know, I mean, it's a toxin. It's a glorified rat poison, essentially, is what it is.
Okay. Okay. So
Yeah.
it's that increase in dosage that it allows you obviously makes it more efficacious.
That is part of it. That is part of what we're trying to convey. So it's the tumor localization, and because it is a prodrug, we can increase the dose to be able to capitalize and get the largest amount of dose directly to the tumor.
Okay. So I read a paper that recently that your group co-authored about inducing a regulated form of cell death in cancer.
Right.
A mechanism known as immunogenic cell death, right? We were just talking. And it's being used as a target to prevent tumor relapse and metastasis.
Yes.
And you point out that although therapies like chemo have been effective in reducing ICD, almost no small molecules have been.
Well,
so how does your innovation compare there?
Yeah, so it's interesting. So the concept or idea of ICD being proposed as an anti-cancer target was probably proposed back in 2014. So it's still relatively young in the grand scheme of therapeutic development. So what they originally did is they took all known, and this is published, they took all known anti-cancer agents pretty much under the sun. They took the NCI, data set and a variety of just anything that they knew that could kill cancer. They evaluated it for its ability to induce ICD. And of those, I think it was at the time, it was over 2,000. I don't remember the exact number, but it was well over 2,000 different small molecules that they evaluated for ICD. So from that study, about 0.05% of those compounds were found to be genuine bona fide ICD-inducing agents. Only 0.05% that were able to generate an immunogenic response to be able to recruit CD8 killer T cells to be able to come and fight the cancer. That's extraordinarily low.
Yeah. Yeah.
And especially for a mechanism that has so much potential with its ability to synergize with checkpoint inhibitors. And in fact, since then, they have published a variety of different reports on true ICD-inducing agents being able to synergize with checkpoint inhibitors. Remember, checkpoint inhibitors are great when they work, but there are obvious limitations associated to them. So luckily, we've been working on this for a while. And so this is nothing new this is nothing new for us. And we've learned a lot of lessons along the way on how to generate ICD-inducing agents. And we have when we've published a few here and there. But the nice thing about the TextCore platform is that we've incorporated or we've made sure to develop agents that have the potential to induce ICD within our platform. So here's the bottom line. So we believe that if you want to kill cancer, you need all the help that you can get,
Yeah.
right?
Okay.
So you want to be able to have something that can, one, kill the cancer, and then if you can actually recruit the immune system to help you out along the way, why wouldn't you do that? And so that's what we've done in a variety of our different discovery assets, is ensured that we have the potential to induce ICD. And that's something that we're continuing to explore.
Okay. So there are, I think, roughly 130,000 ovarian cancer cases that meet the criteria, right?
Mhm.
Are there any competitors that you're aware of going after that market in the same way?
Oh, sure. So I'm going to pass this over to Krystle so she can chime in here.
Yeah,
Okay.
so I just want to give a little bit extra background on the ovarian cancer market. So ovarian cancer is the #1 cause of gynecologic mortality, with an estimated 300,000 women diagnosed globally, of whom 80 to 90% will develop platinum resistance. So we anticipate 100% market capture of rare disease subtypes, as well as a percentage of market infiltration in both wild type and mutant P53 subtypes, which further allows us to respond to an urgent and unmet clinical need by taking advantage of an expedited regulatory approval pathway in ovarian cancer as an orphan disease, and then expanding into other solid tumor indications. Now, we acknowledge that we are not alone in development. There are several early stage companies focused on ADCs for DNA damage or even immunotherapies and CAR T. However, there are significant challenges with tumor trafficking, migration, and immunosuppressive factors in the tumor microenvironment that can become problematic with these various modalities. So we believe this is where InnovoTex is significantly differentiated. From a modality perspective, we are a Texafrin drug conjugate. This gives us a potential edge, as Texafrin has been clinically validated in over 1,000 patients to localize to solid tumors and was well tolerated. We're also not in the crowded market of high-grade serous ovarian cancer. Instead, we're targeting an estimated 35% of those ovarian cancers that are wild-type P53 rare disease subtypes of ovarian cancer that are de novo refractory. And to our knowledge, no one else is targeting these subtypes.
So where are you? What are the plans in terms of getting oxalotex into the clinic? And what might be the perceived challenges in terms of translation to humans?
So we're planning an innovative phase 1A dose escalation design for platinum-resistant ovarian cancer patients, as well as other advanced solid tumors. Based on the published clinical reports from the prior phase 3 studies, there were no grade 3 or grade 4 toxicities that were observed in any patient that received the original Texafrin. In addition, our pilot 14-day repeat dose toxicity study in mice leads us to suggest that bone marrow suppression may be dose limiting in the case of oxalotex, yet this can be managed clinically.
So it all sounds very exciting. It's interesting in that we are recording this podcast during I think it's International Women's Month, and you know, a spotlight's been cast recently on women's health. Can you elaborate on some of those initiatives and how they might impact your work and your company?
Oh, absolutely. So as we attend some of these various conferences, we see a further push regarding the impact of women's health initiatives. And this is most recently emphasized by the White House and Dr. Jill Biden's initiatives on the same topic. To emphasize our marketing reports, we also reference the Silicon Valley Bank report released late in 2023, which focused on innovation in women's health. Now, the report analyzes trends in market and investment strategies across health tech, diagnostics, medical devices, and biopharma, and reveals sort of an optimistic outlook towards a rapidly changing landscape in women's health. Now, since 2019, advancement in women's health has largely been led by the health tech sector, comprising almost half of all women's health deal flow. More recently, however, biopharma has emerged as the top subsector for VC investment over the past 4 quarters, increasing that deal flow to almost 66% of VC investment in the US and Europe, with an average investment of about 182 million per quarter. So there's a significant shift in moving towards biopharma in women's health. For ovarian cancer, as we mentioned, the median survival rate is estimated at 5 years post-onset. It's undetectable and often presents as advanced disease. These platinum-based drugs are widely used as either first-line, combination, or adjuvant therapies. But again, 80 to 90% of these women diagnosed with advanced disease will develop platinum resistance. Traditional chemotherapy is significantly limited due to poor tumor localization, mainly devastating side effects, and then inconsistent efficacy. The median age of diagnosis for ovarian cancer is typically 63 years. Yet for low-grade serous or other rare cancer subtypes, the median age drops significantly to 45. This diagnosis can be years after vague symptoms present. Current standard of care is platinum treatment with carboplatin or cisplatin chemotherapy, yet fewer than 15% of patients respond to subsequent chemotherapy. So we need to not only shine a light on women's health, but actively invest in oncology therapeutics that will increase survival and improve quality of life for women.
Well, I congratulate you on your research, and also applaud your work to help out in terms of women's health. I'm sure that's so important to you. I think it's exciting that we now have, and of course we have had, this has all been going on in the background, of course, for some years now, as you explained, but that we now have perhaps a significant improvement in terms of drug conjugates, delivery, and that sort of thing, over the ADCs that everyone is so excited about. Something more to be excited about is always a great thing to bring to the podcast. So I thank you so much for your time and your research and work, and I wish you all the best of luck. Thank you so much for joining us.
Yeah, thank you so much for having us. Appreciate the time.
We appreciate it. Thank you so much, Tim.
Thanks for tuning in to the Life Sciences and Biotech podcast. We'll see you in the next episode. The information contained in this website and podcast are purely informational and not considered investment recommendations. Tim Doherty's participation in Biotech Insights is separate and apart from his role as an investment advisor representative. Nothing contained herein can be construed as a recommendation or endorsement of any of the companies discussed. Tim Doherty also has no financial affiliation with any of the companies mentioned in this communication. Tim Doherty makes no representation that the information contained in this material is accurate and is under no obligation